Hi everyone, I'm Alyssa and I'm a recent Master's of Biology graduate from Laurentian University and a research assistant at the Northern Ontario School of Medicine Research Lab. Today I will be presenting on the molecular mechanisms involved in radiation-resistant breast cancer. I have no conflict of interest to disclose, and the funding sources for this project can be found at the bottom left of my poster. Breast cancer is the most common cancer in women, and while over 80% of breast cancer patients being treated with radiation therapy will see a benefit, there still remains a good portion of patients that suffer with recurrent radio-resistant cancers. With 1 in 8 women having a lifetime risk of developing breast cancer, this is an important area of research, and we are investigating the molecular mechanisms that promote radiation resistance to identify potential therapeutic targets for these difficult-to-treat radiation-resistant cancers. To do this, we are using a CRISPR-Cas9 system that activates gene expression rather than the typical CRISPR-Cas9 knockout model. As you can see in the top middle panel, we have successfully established MDA-MB231 breast cancer cells which stably express the Cas9 protein. Now, what is really interesting about this project is that we are taking a whole genome approach to overexpress almost every gene, 18,885 to be exact, in the hope that we can find key master regulators of radiation resistance. After performing optimizations to determine how best to infect our cells with this pooled CRISPR activation guide RNA lentiviral library, we have now successfully established a pooled population of MDA-MB231 mutant cells, with each cell having a single gene overexpressed. We then subjected these pooled mutant cells to a repeated high-dose radiation regimen to scan for populations that are resistant to radiation. The next part of this project will be to use deep next-generation sequencing to determine which guide RNA sequences were present in cell populations that had survived this radiation regimen. From there, we will be able to identify a list of genes that confer increased resistance to high doses of radiation in breast cancer cells when they are overexpressed. Thank you very much.